Hi, everyone. My name is Jessica Liu. I'm one of the organizers of the BECS working group. BECS is the Benthic Ecosystem and Carbon Synthesis Working Group. We're a working group with the U.S. Ocean Carbon and Biogeochemistry Program. Our group is aimed at integrating observational data on benthic fluxes and organisms to create data sets that can inform future model parameterization and validation. We're aimed at building capacity in benthic modeling and integrating, the pelagic and benthic biogeochemical cycles. And as part of our small group activity, we have organized a bi-monthly webinar series ranging on focus on a range of topics. And today we're happy to welcome two speakers on benthic oxygen. So our first speaker is Dr. Matt Long from the WHOI Oceanographic Institute, and he will be speaking to us on benthic oxygen metabolism, how technology, physics, and fluxes advance marine biogeochemistry. So Matt, do you want to go ahead?
Thanks a lot for that, Jessica. So I'm Matt Long. I'm from the WHOI Oceanographic Institution in our Marine Chemistry and Geochemistry department. So a little motivation about what drives my interests and what drives sort of the engineering that occurs in my lab. Here we have a very simplistic diagram of a benthic water interface. Here I'm showing seagrasses and corals. I commonly work in shallow coastal environments, but these same principles and physics carry over to just about any benthic environment. So we have some gradients. Those gradients change very quickly at the sediment surface. So it's quite an interesting place to work, but we can also use these gradients and changes to actually measure fluxes. So in shallow water environments, you have light. A lot of that light is absorbed right at the sediment surface by organisms. We have flow profiles that we use the physics of those profiles to actually calculate fluxes. Oxygen very quickly gets consumed as you get near the benthic boundary layer and then quickly goes to zero in the sediments. And on the other side of that, you have nutrients, which are generally higher in the sediments and much more static and lower in the water column itself. And then, of course, you have other processes like microbial and phytoplankton respiration and photosynthesis in the water column, as well as air-sea exchange. So we can use sort of all these exchanges, all of these gradients to actually calculate fluxes and measure those interactions between the benthos and the water column above it. So in my lab, I do a lot of technology development, a lot of platform development, from things like automated bubble traps to measurements of light fluxes to air-sea exchange. But today, what I'll talk to you about is in the red box here, specifically looking at some of the technical challenges and advancements we've done with eddy covariance, as well as moving forward into real-time continuous fluxes. So boundary layer exchange is really the main physics and principles that we're using here. You can think of this in a few different ways. In the video on the left, this is a video from Matt Rodenbach where they put fluorescent dye over coral heads and measured the exchange of that dye in a flume. Here you can see there is both forward currents as well as waves on top of that. So in eddy covariance, we measure at one very specific point in the water column. And you can see that if we measure the current velocities at that point, as well as the sensor concentration, here you can think about it as the fluorescein dye itself. We can take those measurements and directly calculate a flux from those high frequency measurements. There's also other physical processes that are occurring here. We have typically a logarithmic flow layer above the bottom. You can use other techniques by measuring the eddy diffusivity as well as those concentration gradients to calculate fluxes with these sort of fluid mechanics methods. So we measure turbulence, sometimes we measure gradients. We have a number of different tools for measuring fluxes. The same principles apply to the benthic boundary layer as well as the air-sea interface. We're developing tools for doing metabolic measurements as well as calcification and dissolution. And again, we do this through technology and engineering development. So a little bit about the history of aquatic eddy covariance. This is a small review I did as part of a paper. The figure in the top left in red shows that majority of eddy covariance studies has been done in less than 5 meters of water, so generally very shallow. Something we're looking to advance and move more into the deep sea to get some of those deep benthic flux measurements that we're interested in. To the right in green, generally these are pretty slow currents, and that affects the physics and how we measure the fluxes sometimes. And in blue on the bottom left, we have the measuring height. So how high above the bottom are we actually measuring that turbulence and high frequency sensor measurements? And generally those have been pretty close to the bottom. And then in the figure on the right, I'm going to talk a little bit about frequency analysis and how we can use analysis in the frequency domain to actually calculate fluxes and remove some of the biases and apply corrections to improve our data resolution. So we have the measuring height on the bottom in the bottom right graph, and we have the frequency of the contributions. So what frequency are the eddies that are contributing to benthic exchange? And you can see that when you measure very close to the bottom, those are generally higher frequencies that are contributing to the flux. As you move towards higher measuring heights above the boundary, those frequencies become much longer. And we can actually use this to better design our instruments and get away from problems like wave bias. So this is what I'm calling analysis in the frequency domain. This is a fairly simple principle. And I show this in the two graphs here. We have time axis on the one on the left, and that's just a frequent concentration or a signal from 0 to 1, so a normalized signal. You can see there's a lot of noise in that data. But what we can do is we can transfer that into the frequency domain. So what are the frequency of those contributions to the signal? And frequency is just hertz or 1/seconds. And you can see that how when you deconstruct that signal in the frequency domain, you get a very different relationship. And we can use this analysis in the frequency domain very specifically for high frequency data to analyze time series as well as give us further information. So in this part of the talk, I'll talk about 3 different measuring systems. You can see them in the pictures on the top. They're all eddy covariance systems that just have different measuring heights based on the different environments that we deployed them in. So sand flats at about 10 meters deep, seagrass beds at about 5 meters deep, and shallow coral reefs in the Florida Keys at about 2 meters deep. And the reason that we have different measuring heights here is because of the different roughness of the bottom, but also as you move higher above the boundary, your eddies also increase in length. And that's what the figure on the bottom, the caricature, shows those eddies as they interact, as the flow interacts with the bottom and creates those eddies. As you move higher up in the water column, the length scales of those eddies increase and their frequency decreases. So they become bigger and they become slower. And we can actually use this to our advantage. So there's a lot on this figure and I'll walk you through it, but know that these are all analyses in the frequency domain. So in the bottom panels, D, E, and F, we have red is our oxygen signal in the frequency domain. Blue is pH or hydrogen ion concentration. Green is our vertical velocity, and that's what we actually use to calculate that sediment water column exchange. And then we also have VX, which is our main longitudinal current direction. And you'll see a dashed line in the middle of that figure, which is FC, or the cutoff frequency. And what you see at higher frequencies above that dashed line is you see wave peaks in the vertical and the horizontal velocities. So those are actual wave orbitals coming through. And if I can draw your attention to the top panels now, this is the normalized cumulative cross power spectral density. So cross power spectral density is analogous, or it's actually the exact same thing as regular eddy covariance analysis. We're just doing it in the frequency domain. So that's what the cross power spectral density is. The power spectral density on the bottom is just the contribution of that signal at specific frequencies. And what you'll see when you look on the left-hand side of that dashed cutoff frequency line is that we have pretty standard contributions to the fluxes, and they follow theoretical estimates, the dashed, bigger dashed dark line being Keimal estimates using the -5/3 slope and fluid mechanics theory to actually calculate what our contributions should be based on a wave number spectrum. And what you can see, for example, in the momentum fluxes and the gray line on the top panels, you'll see that at the wave frequency, you can have very large contributions to that momentum flux, which is caused by wave bias. You can also see that in some cases in the oxygen and in the hydrogen ion concentration. And these are actually very nice spectra that I'm showing here. There are other spectra where there's significant contributions at wave frequencies, which is biased in our flux signals. So I can go into extensive detail on this, but I just wanted to give you a quick flavor of how we can use this frequency domain analysis to get rid of wave bias in our signal. We can use this cross power spectral density to calculate eddy covariance fluxes in the frequency domain. And we can use these cutoff frequencies to remove those high frequency variations while still being sure that we're capturing the majority of the flux signal. So from these three sites, this is an example of summary data, and this is from my eddy covariance hydrogen ion and oxygen exchange system, or ECHOs. So in the top two panels, we have the oxygen flux, and that's across 24 hours. So this is a number of days of data averaged hourly together. In the red coloration, we have the sand fluxes. They're obviously the smaller fluxes. In green, we have the seagrass bed. You can see the fluxes are much bigger there. And then the gray bars are the reef. And I should highlight that these are fairly degraded reefs in the Florida Keys. So these are dominated by octocorals, sea fans, sea rods, that sort of thing. And you can see there's a very strong net heterotrophy in those degraded reef environments. So moving to the top panel on the right-hand side, these are the cumulative fluxes. So what is the actual net rate over a 24-hour period on these? As you would expect, the seagrasses are net positive oxygen producing, consistent with the fact that they are carbon sinks. Interestingly, the sand is also net autotrophic, and that's because if you look at the picture in the top left, you'll see there's a lot of microalgal mats growing on those bed forms. And then the reef, again, here you see that very strong net heterotrophy. Then in the bottom panels, we have a hydrogen ion flux, which is a fairly new metric that we've been working with. You can see those diol fluxes, they mirror the oxygen fluxes very closely. But interestingly, in the cumulative fluxes on the bottom right, we have this degraded reef environment being a very strong source of protons, so a very strong acidity producer. The sand is about balanced, and then as you would expect with the seagrass beds, they are a net acidity sink. So this is just kind of an example of how we can do this frequency domain analysis. We can remove wave bias and really get these high quality hourly fluxes out of the environments that we're interested in understanding that benthic water exchange. So great, we can do this frequency analysis, so what? Well, it really removes wave bias, which has been a very challenging aspect of doing aquatic eddy covariance, especially in shallow systems. This also allows us to do measurement height recommendations. So how high should you sample based on the peak current velocity, the wave periods that we expect, and how can you tune your system to actually sample at the right height to enable that wave bias removal. The other sort of side effect of this is it really enables new sensors. We've been extremely limited in eddy covariance what we can actually measure because we need sensors that can measure at about half a second response time, and that's very challenging to produce a sensor that does that. But now that we're using this cutoff frequency and removing things, we can now use much slower sensors. So a sensor that responds in 5 seconds or possibly even 10 seconds, depending on the conditions at a site. So we can also remove any wave sensitive sensors. So by doing these cutoff frequencies, you can remove problems with sensors that are sensitive to waves. And we're also now packaging our sensors in flow through paths. So we can now utilize sensors that are also light sensitive. So these are kind of benefits of being able to do this analysis in the frequency domain. And I'll talk about a couple of other advantages going forward. But now I'd like to move into the second part of my talk, which is focusing on really what our long-term goal with eddy covariance is to enable these long-term flux measurements. And the two figures that you see on this graph of the world are all terrestrial eddy covariance towers. So these are terrestrial eddy covariance towers that are measuring fluxes of CO2 primarily, but also water, temperature, and a variety of other things. And that's simply because the technique in the terrestrial boundary layer is much more developed. But what you see here are actually continuous flux measurements that are being done throughout the terrestrial biosphere. So the question for our technology advancement is, how do we enable that underwater? And the pictures that you see in the water here are a buoy and lander system that we've developed to enable these long-term flux measurements underwater. So this project gets started in the middle of COVID. My postdoc, Jeff Coogan, on the left there, started a week after WHOI shut down. So we had to figure out how to enable building this whole system while we were at home. So I sent Jeff off with a whole bunch of equipment. I went to my garage and started welding things together, and we ended up with this buoy system that allows for continuous power via solar, as well as real-time communications, so we can understand how the system is doing and if we need to service it. And we deployed this system right outside of Woods Hole in a shallow eelgrass meadow. So here you can see pictures of the buoy lander system. There's a picture of the lander underwater. You can see we're in very green, sort of high productivity temperate waters. There's a lot of fouling that occurred, which is one of the major challenges we all deal with when we try and put things long-term in the ocean. But basically a fairly complicated electrical architecture with the instruments on the bottom, including acoustic Doppler velocimeter for measuring turbulence. We used Honeywell ISFET pH sensors to measure the pH flux. We had a Rinko DO and temperature sensor to measure oxygen flux. We had a number of pumps, a back flushing pump to remove any fouling that occurs at our system intake. We had a controller that everything plugs into. We have surface measurements of PAR, wind speed and direction, and then solar power, of course, battery systems, and then we use cell communications because we're in the coastal zone, and that communicated to a remote server at WHOI. And we were able to deploy this system from the beginning of May into November, and the first time that we tried this. You can see the water temperature in the top panel here over that time period from the system. We also did a lot of measurements in the seagrass bed itself, looking at seagrass blade length and other metrics, but that just allowed us to get a picture of the biomass as well as the algal biomass and epiphytes on the seagrasses in the bottom panel there. So we kind of get this full growing season picture of what's going on with the benthic fluxes. And here's what that looks like. The top panel here, you see a couple gaps that occurred throughout the year, and that's due to servicing or changing the sensors that we needed to do. We were kind of learning along the way. We've never deployed anything for longer than about 2 weeks before, so this was a big jump forward for us. What you see in the oxygen signal is it starts relatively high with low variability, and that's low water temperatures causing the concentrations to be higher, but also less productivity at those times of the year. As you move into August and September, you see the oxygen concentration goes down, simply a function of temperature, but also that daily variance increases substantially due to primary production and respiration when the waters are warmer. And then going into the fall, you can see the variance again decrease. You can see the temperature in the middle panel. We can also do temperature fluxes directly with this eddy covariance system. And then in the bottom panels, we have the oxygen flux, and the blue bars are the daytime measurements, the red bars are the nighttime respiration measurements, and the black line is our net ecosystem metabolism that we can integrate over this entire time series. So part of this is, again, using this analysis in the frequency domain. So we could look at not just what's going on in the frequency domain, but we can also use it as a metric for understanding how well our sensors are working. So in this case, we have oxygen power spectral density on the right and our temperature power spectral density on the left, and on the right, we have the temperature power spectral density. And that temperature sensor is less prone to fouling. It's just a little metal rod sticking out into the water column, whereas the oxygen sensor has a sensing film that can become fouled and slow in response time over time. So in the blue lines, when we first put the system out from day 1 to 30, you see that we have a very nice response. If we look at day 30 through 60 in the red line, you see that we have a reduced response at high frequencies. We also see the same at purple at day 166 to 176. What we did at day 136 in the yellow line was we actually cleaned our sensor off, removed all the biofouling from it, and we see that that response time returns again to what we got at those very first days. So this one gave us an indication of how often we need to service our system. We've implemented wipers onto our sensors now, and really we can get some pretty cool data out of this. So here you see curves from May through October, and if you follow them up, if you see my cursor here, May is down here, we go into June, we go into July, and then peaks in August, and then comes back down in September and October. So these are oxygen flux versus light, so a standard photosynthetic PI curve. And then we can also look at hour of day to see how those curves change throughout the year. Again, May, June, July, August, all the way up here at the top, but the maximum variance, and then coming back down in September and then October. So we're really just scratching the surface of what we can get from this long-term flux data. We're kind of overwhelmed with the amount of data we have, but many more results and papers coming from this data set. So just quick takeaways from what I did. You know, we get the fluxes, which are the numbers we need. We can use this frequency analysis. We've got this surface buoy that allows for real-time communication and analysis. We can analyze in real time how our sensors are working. We're using a variety of tracers. We're using oxygen, momentum fluxes, pH fluxes, and temperature fluxes. And now that we can utilize slower sensors, I'm very curious and excited to talk with others about what other new sensors we can implement. And then, of course, pairing this with other technologies for doing air sea fluxes, water column rates, as well as things like bubbles. So that's all I had. That was unfortunate that my computer died halfway through my talk.
So Matt and I have been in conversation for quite a while about a potentially very important role that he and other benthic geochemists could play in getting more of a synthesis established between water column and sediment geochemists to studying important problems, particularly in the ocean margin setting. And our dialogue came up a little over a year ago, and I proposed that we get some type of margin basin exchange initiative going, which might be both a kind of initiative that geotraces might transition to, but also a way of bringing the benthic community more into the emerging biogeoscapes program that Heather was talking about. And so I got a group of about 15 people together from the modeling, sediment, and water column communities together. We talked about some important processes that might be important in our particular area that we highlighted, which was the exchange of materials between the continental shelf and the interior of ocean basins. So cross-shelf exchange along continental margins. I got some considerable amount of feedback from Hetty Edmonds at NSF Chemical Oceanography about this, some of the most important of which was she felt strongly that I should get the carbon community more engaged in this. So I was delighted when Matt invited me to this group. I'm also a sort of subtext of this. I'm very interested in capacity building within our community, particularly in the area of sedimentary biogeochemistry. And so I was delighted to have an opportunity to speak here. I'm particularly interested in the fact that I only know or am familiar with about a quarter of you, which I think speaks a lot about the success of Heather and the other organizers in getting a kind of an exciting group together. So these are kind of the things that we were focusing on: margin basin exchange processes, the role of subsurface currents and eddies. And here I'm talking about eddies at the sort of sub-mesoscale level. So not in the sort of things that are important in cross-shelf exchange, but these would be of materials that were mobilized by the much smaller eddies that Matt has been talking about. And in contrast to geotraces, which has a basin scale objective, looking at more of a regional focus. So I want to talk a little bit about the physics. This is a conceptual diagram in a paper by Frenger et al. That was published a few years ago. A lot of new terminology was introduced here, particularly eddies that are derived from the interaction of undercurrents that flow along the continental margins with seafloor topography. And at this point, we have to get away from the idea of the continental slope and this shelf slope break as being these kind of uniform planar surfaces, and think about the details of topography, particularly in places like the west coast of North America, where there are very strong eddies generated by the very rugged and exciting topography along the continental slope. So basically, what happens is that these eddies are generated by the interaction of, in the case of North America, undercurrents flowing in a polar direction. These eddies are generated and move offshore. Some of them also move onshore. But what this can lead to is a net cross-shelf exchange from, as I mentioned, materials mobilized over shelf sediments and then advected offshore. Eddies that are generated by poleward undercurrents are often referred to as putties. I'm not sure if that terminology was coined in this paper or elsewhere. But what you see within these eddies is transport of material, which includes a microbial community that's composition was derived from the shelf, chemical constituents, and so forth, being moved offshore and often retaining coherent properties. And as I mentioned before, terminology like stew pot and cannonball transport were coined in this paper. I'm not sure how widely they've been adapted, but they are quite useful in describing the concepts. These are some simulations here that were shared with me by my collaborator, Daniele Bianchi from UCLA, showing how these eddies affect the exchange of oxygen and nitrous oxide between the eastern boundary upwelling system off of South America and the interior of the basin. And what one can see, particularly say focusing on the oxygen at the left, is that there's a lot of, this is a very dynamic system. These eddies result in, as I mentioned before, transport, in this case of cold upwelled water, offshore, but also the entrainment of eddies that move in shore as well to replace some of these materials that are moving offshore. But of course, the thing that interests us as geochemists is the overall rate of exchange and how this can provide a vector for cross-shelf exchange that can lead to significant changes in the composition of the water column of the interior basin. How do modelers like Daniele, sediment geochemists, and water column geochemists actually get together to compare some of their data sets? And I'd like to provide an example based on the Peru system. And this represents an interaction between data sets generated by Greg Cutter on the Geotraces GP16 cruise, looking at excess iodide, which accumulates in the water column as a result of high benthic fluxes. Florian Schultz from Geomar in Germany, who actually measured benthic iodide fluxes on the Peruvian shelf, and Daniele, who with his postdoc Simone Yang applied the model that I just showed you to the offshore transport of excess iodine. And basically, he was able to simulate the type of transport that we were seeing given these high benthic fluxes. Although the real data indicates that the excess iodine was much more sharply confined to an isopycnal that covers a rather narrow range of densities shown here that cover a fairly large area of the Peru shelf. So there might actually be more stratification in the real world than is predicted in this model. Now, this is what I would call an after the fact interaction, in the sense that this was Daniele applying data from geochemical measurements from cruises that had already taken place. But I would argue that moving forward, and something I'd like to engage this group on, is that we really need to engage ROMS modelers like Daniele from the inception of these projects in terms of guiding our future work. And this is important for a number of reasons, but largely because we need their insight into sampling strategies. This is a data set that was compiled by one of Daniele's students, Danny McCoy. And this is his first paper he published in Progress in Oceanography, which is a pretty ambitious move for a graduate student to publish a weighty tome in that journal. This is not model data. This is actual real data for some of the eddies, which are designated here as submesoscale cyclical vortices, or SCVs. And what they represent are observations of salinity and temperature taken from a vast amount of Argo float data and interpreted using some algorithms that Danny applied to actually look at these features as they move offshore. The most important take-home message from this paper is that these features can move materials a long way offshore. So when we're talking about cross-shelf exchange, we don't really mean impacts on the slope. We mean impacts across the entire basin. So I think this is an exceptionally valuable data set in giving us an indication, but also maybe giving us some insight into how we should conduct some of our field campaigns. This is a simulation here from another ROMS modeler, Misumi, in Japan, which was published several years ago. This is a simulation of surface water dissolved iron. This iron, according to the model interpretation, is generated largely within the Sea of Okhotsk, north of Japan, and to the east of Sakhalin Island, which I'm running my cursor over right now. It's advected to the south through a variety of channels along the Kuril Islands here, and then makes its way into this big plume that gets entrained in the Kuroshio current, an important western boundary current, and moved offshore. The model, intrinsic to this model, is the role of eddy-mediated transport offshore. And it's interesting that this area corresponds with a variety of, or a high density of minty submesoscale cyclical vortices identified by Danny in his data set. They're indicated in blue here. And the blue or minty SCVs correspond to eddies that have a lower salinity and also lower temperature than waters of the same density surrounding them. And it's pretty clear that these are probably coming from the Sea of Okhotsk based on some independent data that Japanese researchers have seen. And this little, these arrows down at the bottom here indicate their average drift, and they become very important at moving these materials offshore. Misumi's model has been very useful at simulating the offshore transport of iron. The observations are shown in the top right, where there is iron basically being ejected quite far to the east. And again, a lot of this is mediated by transport from the Kuroshio current. There is also you can see a big broadening of this feature that's associated with sinking particles. That process is hard to measure. What the modeler figured out is that the best way to parameterize that most successfully were simulations E and F in this figure. I don't want to go into that too much. This is a very exciting aspect for people who study iron in the interior of the basin. But I think what I wanted to point out here is that the sort of the booster, if you will, that got iron into the basin in the first place and injected it into the Kuroshio was the eddy-mediated transport off of the Kuril Islands, incorporation into a southward flowing current called the Oyashio, and then getting this material into the Kuroshio. If we look at the entire North Pacific basin, what we see is this massive plume coming from the west off the coast of Japan, but we also see a big plume coming off of North America, not extending as far to the interior, but also being a very important feature. So this leads to the question of what might be some of the dynamics associated with that plume. And here I really want to get to the gist of why we need to have a modeling community getting us some insight into how to go about conducting field campaigns in the most effective way. First off, before I actually explain this figure, I want to tell you a little bit about ROM's models. These ultimately are physical models based on primitive equation models. And one of their key features is that they have very high resolution, often 1 kilometer by 1 kilometer or 4 kilometers by 4 kilometers, that enable them to look at realistic features of bottom topography. One of Daniele's postdocs, Pierre Damien, generated this simulation for us, which actually looks at, in this case, iron flux across the shelf and into the basin, with the areas in red being offshore transport, in other words, to the west across, in this case, the Oregon shelf, and the blue being onshore transport. And these are not random features. These are mean fluxes over an entire yearly cycle, and they are set by specific features in the seafloor topography. I should mention, of course, that we have a focus on iron because that's what I do, but also because we're very interested in sources of iron which can limit primary production in the North Pacific. This is not something that you just see off of Oregon. There are similar features off of Peru, and down in Peru, they're so persistent that a German physical oceanographer, Lothar Strama, has actually assigned names to the currents that are generated by these types of features. And a physical oceanographer only assigns a name when something is a persistent feature. Why is this important for me as an experimentalist? Well, it gave me a pretty good idea of where we ought to be sampling if we want to see places where transport of iron and other materials offshore really matters. Now, I want to talk a little bit about how somebody would make a transition from tracers like nitrous oxide or oxygen that I showed you earlier and something like iron. And a big part of this is you have to model the source. And a fundamental assumption of the ROMS models that have been used to, and actually other models of iron in terms of sedimentary sources, is that bottom water oxygen is the key determinant of iron fluxes. And this is a redox related phenomenon. Reduced iron, iron 2, which can only accumulate when oxygen is low, is much less particle reactive than iron 3. So reducing conditions provide an escape mechanism, if you will, for iron to get off the seafloor so that it can be advected away. These data here are benthic flux measurements, the kinds of things that Matt does. They're not necessarily water column measurements, but they do show that there's this kind of threshold. And notice that the scale here is kind of in the reverse direction: high oxygen on the left, low oxygen on the right. And the oxygen has to be less than about 60 or 70 micromolar for these fluxes to be high. Now, an important thing about all of these ROMS models is that they're really not about iron. They are, what they really model explicitly are oxygen consumption rates associated with carbon oxidation and remineralization, both within the water column and at the sediment water interface. And so when you see one of these systems modeling iron, it's basically not really, well, it is modeling iron, but the driver of the source is the carbon and oxygen cycle components of that model. And these are some data here generated by On Pham, another postdoc with Daniele, who is in On is a he actually is an iron expert. He knows a lot about iron and has had a lot of experience working with iron. And these are some data that he generated recently for a paper he's been working on along with Pierre and of course Daniele. And the gist of what this thing predicts is that you have, if I would start with slide B on the right, you have cross-shelf velocities near the seafloor. And what we care about here is between, say, 175 and 200 meters. So water's overlying the outer continental shelf off the Oregon coast. And some of the highest cross-shelf velocities occur in the late summer and fall in this area. You can also see the strong seasonal upwelling near the surface. That, of course, also leads to offshore transport, which is in the red. And then we have our sort of canonical Ekman transport with onshore transport kind of in this vast blue area in the middle. So that's panel B. Now, the interesting thing about that is that maximum in cross-shelf velocity also coincides with a minimum in bottom water oxygen, because as many of you may know, the Oregon coast is very well known for generating a lot of hypoxic zones in the summer. That low bottom water oxygen leads to a significant release of iron from sediments, a high flux. And so we have this kind of a sweet spot of the highest iron fluxes being associated with the highest cross-shelf velocity. So guess when I decided to stage my cruise, wanting to capture this event, was basically in August and through into mid-August, so that I could basically get, and we staged this cruise in 2021. And the other thing, of course, is if I wanted to control when these fluxes would be low, the best time to go would be March or April, and we did that as well. Moreover, cycling back to what Daniele had, or the insight we gleaned from Pierre, we knew where we ought to go. And in this plot here, there's obviously a transect or map of the Oregon coast on the far right. You can see that in the left, there's, I want to basically turn your attention to the whole combination of factors that I just showed you previously. What's remarkable, if you look at panel A and you look at the red line, the offshore transport of iron is defined by that red trace. And what you can see in On's simulation here is that most of the iron transport off Oregon that we think contributes to that big plume in the data that I showed you previously comes out through a few places, sort of choke points, if you will. And these are driven almost entirely by the interaction of the poleward California undercurrent and specific features of the undersea topography. So if you want to understand fluxes, you really need to work with a modeler right from the get-go so that you know where to conduct your measurement campaign. And I think this is exceedingly important, not only for somebody working on iron, but for carbon as well. I think this is the type of approach that would be very useful to looking at carbon and oxygen budgets across continental margins. Much of our field campaign was directed at validating the main hypothesis or the main assumption that went into linking iron with what is essentially a carbon and oxygen model, which is that bottom water oxygen is the big factor. These are just plots here showing total dissolved iron and iron 2 at a number of stations up and down the Oregon coast. And what they show is that the total dissolved iron and the iron 2 really only get very high when the bottom water oxygen shown in the panels at the bottom is low. And in some cases where the oxygen is really low, the predominant form of the iron is iron 2, which, as I mentioned before, is highly soluble. It really promotes the offshore transport of the iron and kind of getting it away. This is a plot here of oxygen and iron 2, kind of a cumulative data set, both from the water column and also within the benthic boundary layer that we got from measurements using a benthic lander, kind of analogous to what Matt uses for his eddy covariance measurements. And that really was an important validation of that critical assumption in the model that bottom water oxygen really matters in terms of iron escape into the overlying boundary layer. But if we were to apply this more broadly, we have to consider whether other factors matter as well. And another important consideration is how much, what fraction of mud is in the sediment as opposed to coarse grained sand. And this gets a lot, this kind of circles back to the fact that carbon oxidation rates, as well as bottom water oxygen can be very important. These models, by the way, explicitly look at carbon oxidation rates because that's a very effective way to study nutrient dynamics in these systems. Carbon oxidation releases nitrogen, for example, from organic matter at the sediment water interface, and that's very important. But you can have a lot of carbon oxidation releasing iron, and if the bottom water oxygen is high, that iron gets oxidized and reprecipitated fairly quickly. That's why bottom water oxygen matters so much.
Hey, Jim, this
yes.
is Heather. It's a little after 1 and our participants are starting to have to go because we.
Yes, I'm sorry about that. Yeah,
and Matt has joined us back too. So I didn't know if he wanted to take a couple minutes to wrap up the last. Sure.
Yeah, I could basically do that. I mean, I wanted to show you that we, in our areas offshore, these hotspots for transport, we actually did find very high rates of offshore transport in agreement with the model. The seafloor topography in these areas from ADCP data really shows that this is the case. And I have a thing about future planning here, which I think is something that I would like to engage people with. But I would be quite happy to get off at this point.
We can probably take a few questions if people are still around.
I had a question for you, Matt.
Yeah.
How big is the eddy covariance community? And do you guys, since it spans all these regions like terrestrial and you know, benthic marine and other aquatic systems, do you guys ever get together at meetings? You know, it's hard to go to meetings sometimes with terrestrial partners, and I'm just sort of wondering how do you exploit whatever critical mass you actually have?
Yeah, the underwater eddy covariance community is very small, right? I think with that like mini review that I did, I think it was like 100 papers, right? So not that much yet on the water side. Terrestrial side, there's over 1,000 real-time continuous towers out there, so we know a lot more on the terrestrial side. And I've tried to make those inroads with the terrestrial groups, you know, because the physics don't change. It's still boundary layer analysis and physics. And honestly, I just haven't had the time to make those connections yet, but certainly on my list of things to do.
Yeah, Jim, you talk about using models to help inform your sampling, but it strikes me that with some purposely placed eddy covariance sites, you could also use that to inform some larger, if you wanted to do, you know, kind of cross-shelf sampling and have some sites there.
Well, I think one thing I want to point out that Matt would probably point out too, of course, is that eddy covariance is very useful when your analyte can be measured in real time with a sensor. When you have to retrieve the sample and analyze it back on the ship, it becomes less so unless you have an analyte that you can measure in real time with a sensor that's a really good proxy. So that is something to keep in mind. But I'm intrigued by Matt's work because I've seen not only his work, but the results of others like Claire Reimer's, that things like oxygen consumption rates at the sediment water interface are really different when you use that method, and they're probably a lot more accurate. So I think that's a really important point.
Hi, yeah. So this is Jerry Wiggard. I'm listening to both Matt and Jim, and my question is, because I've gotten sort of into modeling on the sort of inner shelf estuarine linkages, and my broader question really comes to the group, but also to both those guys, is the connectivity of benthic interaction introducing nutrients into a system, and then how it influences primary production in the euphotic zone. Are we making these linkages here? I mean, I know there's like interest in nutrients and whatever chemistry going on at depth. But how are we translating all of this into how does it impact primary production? And I'd love to hear responses out of Jim or Matt on that.
Well, I guess the first thing I would say is that the model that Daniele and his group are using originally came from a study Daniele was involved with, led by Curtis Deutsch.
Right.
And that model paper was published in, I think, 2020, Deutsch et al. And that was explicitly looking at basically at nitrogen versus iron limitation of primary production in the Northeast Pacific. And so I think most of the advances that have been done since then through Daniele and Pierre have been to refine the iron aspects of that model. But it was based on I mean, that was the objective of that model, and that continues to be an important overall long-term objective of the people working on this.
Yeah, and to expand on that a little bit, you know, we're developing tools to measure benthic fluxes, as well as we're developing a class of automated water incubators so we can understand what the pelagic metabolic rates are. That doesn't get us to iron or nitrogen limitation. There we need somebody like Jim that can come out and make those measurements side by side with our flux platforms that actually figure out what those linkages are, and then we can scale that with modeling.
Yeah, and I guess so. Follow up for Jim. You know, we get this the impact of low oxygen environment introducing dissolved iron into the system as it oxygenates. You know, as you get sort of mixing and what happens to that iron? I think I think you and I have talked in the past about it precipitates out, but how much of it is there any sort of contribution to releasing iron limitation if that is present in any given system? I think it's been a while since I asked you that question, so maybe there's more to how that's evolving.
Yeah, well, I think I think some of the most interesting aspects of that are coming from our colleagues in Japan, where I happen to be right now, because the Kuroshio really pushes a lot of those iron-rich waters from the Sea of Okhotsk into the interior of the basin. And there is, removal due to geochemical processes, but there's also a lot of release of particulate iron, and that enables much longer transport than we might have thought of previously. Everybody does a lot of, has been focused historically on dust when it comes to iron limitation, but the margins are turning out to be highly important. And I also want to circle back to the fact that, as I said before, these models are all basically carbon and oxygen models. So for those of you in this audience who are really focused on carbon and oxygen, they have a lot to offer.
Yeah, I think I think that these models are very powerful. I should also note that the model that Daniele uses, if it's the one that I'm thinking of, it's the ROMS and BECS model, does not have any benthic dynamics at all. And so that's one of the primary missing gaps in a lot of these biogeochemical models.
I should also point out, On's here, he would probably agree with me, but the river contribution needs some work as well. And I think that, one thing where I think OCB could play an important role, having already been through this exercise of working with wetlands and carbon budgets and in those areas, is maybe talking a little bit more about the river continental margin interface. So that's something that I think we should be thinking too, but definitely, yeah, in terms of benthic dynamics, that would be great to figure out that the slide I showed at the very end, which was like the mud versus sand thing. That's like a first step. But there's not a lot of places around the world's continental margins where there's even that much data. So that can be a challenge as well.
Matt, you talked about your ultimate goal is to have a big network of these in situ measurements. Do we have a sense from things like these ROMS models that Daniele's using, how many of those systems would we need in order to, be able to scale up and characterize to some degree the fluxes in these continental margin systems? Is there an approach here? Are we looking at, sentinel sites or areas of bioregions, ecoregions? What do you have any general sense of how many of these systems you would need to develop and place in order to characterize these dynamics?
Yeah, that's a great question. I think Jim brought that up. Sampling at the right places are key, and I think we can intelligently form that with existing data and models. Is the technology ready for that? Probably not. How did it advance in the terrestrial boundary layer? Well, Lycor started making mass available units that you could buy for 40 or 50K. And we're simply not there on the water side yet. Sort of as an underwater aquatic eddy covariance community, we haven't really decided as a group how to do these measurements the best way. I think there's still some technology advancement. We're going to need some commercial partners, and then we're going to need models and observationists to tell us where to put those. So it's coming, but it's a little bit down the road. And, I think we're making those first steps with long-term deployable systems like I showed today. So we're getting closer, but it's a long road.
Well, thank you both. My internet's about to drop again because the cable person is here to fix things. And the first thing they have to do is make it not work for a little while. So, but thank you both for these fantastic talks. And thank you, Chris and Jessica, for all of the work that you're doing to organize these great lectures.
Yeah, thank you so much. And I'm sorry about well, thank you, Jim, especially for joining us from Japan, and Matt, we're sorry that you got cut off halfway through your talk and wasn't able to finish. But yeah, this was a pleasure. Yeah, I'm looking forward to kind of hearing a bit more as we go along in this webinar series about benthic oxygen dynamics. But next time we will focus more directly on benthic organisms and their contributions to biogeochemical fluxes.
Night, Jim.
Thanks.